Hi, everyone. Welcome to STAT's virtual event, ASCO Recap Live, sponsored by Precision AQ. Thank you all for joining us. I'm Catherine McPhail, programmer of editorial events here at STAT, and I'm sure you all know how these things work by now, but before we get started, I have a few quick housekeeping items. So at the bottom of your screen, you will see several icons. You can use the chat feature to chat amongst yourselves and to send messages directly to the STAT team. Please do not put your questions into that chat. If you do have a question that you would like to ask us during the event, please use the Q&A feature, and we will try to have those questions answered live. In the Q&A window, you should also be able to see the questions that other attendees have asked, and you can upvote the question that you would like us to answer. We will be taking questions throughout the session, so do not wait until the end. We really want to hear from you. That's what makes these things fun. Thank you all for being here. Before we get started, I would like to welcome Ashwin Autry, Executive Vice President of Strategy, Innovation, and Partnership at Precision AQ, for a sponsored introduction of today's event.
Thank you, Catherine, and good morning, good afternoon, good evening, based on where and when you're tuning in from. And after an invigorating few days in Chicago with over 40,000 attendees, I'm really excited to share a few thoughts on ASCO 24. To do so, I wanted to start with a few key trials that really stood out. And something that really stood out was the term practice changing, especially with many targeted agents. And the three that I would really like to highlight, along with the continued discussion after this, was the 5-year CROWN trial data. And this is where lorlatinib continued to show progression-free survival versus crizotinib in patients with previously untreated ALK-positive non-small cell lung cancer. And here, the median PFS of patients had yet to be reached. This corresponded to the longest PFS ever reported. And interestingly, the majority of patients, over 92% assigned to the lorlatinib arm, were free from intracranial progression. The second one was the LAURA trial with the EGFR TKI. It demonstrated significantly improved PFS over placebo, but 39.1 to 5.6 months in a phase 3 trial for EGFR mutant non-small cell lung cancer following chemoradiotherapy. And this was the first time an EGFR TKI showed such a benefit in this setting. And actually, it was hailed as most likely to provide a new standard of care for EGFR mutant diseases in this setting. The third one I'd like to point out, which is the talk of the town, was the phase 3 Destiny Breast 06 trial, establishing the ADC trastuzumab deruxtecan as an effective new treatment option that significantly prolonged progression-free survival of patients with HR-positive, HER2 low, and HER2 ultra low in metastatic breast cancer following one or more endocrine-based therapies. Along with these and other results, something that was almost a theme was a push towards earlier treatment settings. And it drives a particular discussion about and brings certain questions such as, how do we actually look to balance toxicity and adverse event profiles with prevention and risk of recurrence, especially in earlier stage diseases, where the toxicity risk may be less acceptable to those patients versus those with metastatic diseases. And especially as we observe the new platforms such as the ADCs with lots of targets and payloads across many tumor types, pushing for earlier line treatment with an AE profile that's a moving target. The second is, how many drug doses pre-surgery are the right amount? Again, a question of when and how. If there's a pathological response, does the patient not receive any further drug? Or do they receive a year of adjuvant drug therapy? And this was really beautifully highlighted in a phase 3 trial with 2 cycles of ipilimumab and nivolumab prior to therapeutic lymph node dissection. It significantly decreased the risk of disease progression and recurrence or death among these patients in stage 3 melanoma. And this allowed about 59% of the patients to actually skip adjuvant therapy. And all this comes full circle when we look at these advancements in treatment options and technologies, along with the potential for earlier targeted treatment in the protocols, highlights the need to better understand the impact that payers and providers can have on patient access to these novel therapies and technologies. And this is where we feel that ASCO's mission and focus is actually very consistent and aligned with the mission of Precision AQ, where through our focus on the balance of IQ, which is the data and the science of cancer care, and the EQ, the empathy and art of cancer care, we hope to achieve our mission to ensure access to life-saving treatments for all. And with that, opening a few statements and highlighting a few key topics that were heard and the drive towards practice-changing innovation, along with earlier treatment setting options being more available. I think that's really exciting. And with that, I would like to hand it over to the staff team to kind of talk through more about the details of some of these insights. Thank you.
Thank you, Ashwin, for the introduction. So here to join me for today's discussion, I have Angus Chen, cancer reporter at STAT, Adam Feuerstein, senior writer of biotech at STAT, Matthew Herper, senior writer of medicine and editorial director of events at STAT. Thank you all for being here. You guys have had one night of sleep at home, so I'm sure you're all completely rested after 4 busy days in Chicago.
Oh yeah, we feel great.
Oh, great. So before we get into the Q&A portion, we are going to quickly recap some of the biggest data from the meeting. Let's see, next slide, please. To start us off, we are going to be looking at Tagrisso. Next slide. Matt, would you tell us a little bit about the data we have here?
Yeah, so this is really cool. These are obviously 2 lines for osimertinib or Tagrisso and placebo, and we're looking at how long it takes for cancer to come back. And as Mark Lewis, who's a wonderful doctor and patient advocate who we all love, wrote on Twitter, the cool thing about these curves is you can drive a pickup truck through them. The issue with this is that there was an 80% crossover, meaning 80% of the people on placebo got osimertinib when they had tumors show up, which may make it hard to get survival results, although there was a difference in survival favoring osimertinib, and those data simply aren't mature. They may actually be mature. This may very well be life extending as well. But I also think one reason that the discussant on this at ASCO brought up that this might not be an issue is the next slide. Could we advance one? So this looks complicated, but I just want to make you to look at that highlighted box, which is the percent of patients who had a new cancer lesion in the sites listed, and that's brain. So you had 8% in the osimertinib arm and 29%. This was also what was seen in the CROWN trial of Pfizer's lorlatinib in a different group of patients. Both of these are genetically selected for a particular kind of mutation. In osimertinib's case, it's EGFR. But like, we're really talking about people not getting cancer in their brain, which is a pretty strong endpoint. So I mean that would be my big takeaway from this trial. The other, which is as a bunch of these studies are, it's an AstraZeneca trial. Really was an AstraZeneca ASCO. I'm sure we'll talk more about that in the discussion.
Thank you for that, Matt. We can move on to the next slide. This is also from AstraZeneca, the Enhertu data. Next slide, please. Adam, will you tell us about the data that we have here? Oh, Adam, you're on mute.
Sorry about that, man. So yeah, this is a study that was conducted by Daiichi Sankyo and AstraZeneca. Enhertu, people probably know Enhertu. It's an antibody drug conjugate that targets HER2, HER2 and targeting antibody. And if you remember 2 years ago, this drug also made waves. Matt, I think, actually covered that 2 years ago when they sort of pioneered the use of Enhertu in patients who have low levels of HER2 expression. Traditionally, these drugs were used in patients who overexpressed HER2, but what we're seeing with Enhertu, with this ADC, is that you can actually treat patients that have much lower levels of HER2 on the surface of breast cancer cells. So that was kind of the big news 2 years ago. This year, this study, what's interesting about this study, and again, practice changing, is this is the most common form of breast cancer, hormone receptor positive breast cancer. These are patients who have already gone through endocrine therapy. And then basically the choice for physicians at that point is, they usually will go to chemotherapy. Usually capecitabine is the drug that's used here. So what Daiichi and AstraZeneca did was randomize these patients to either get an HER2 or physician's choice of chemotherapy. And as you see here in the study, this is a tumor progression slide, obviously a significant benefit with a hazard ratio of 0.62. So you've significantly reduced the risk of tumor progression. Now, what's also interesting about this study is what we're seeing here, these are in the HER2 low patients. These are patients that have low levels of HER2. They also tested this in this study in a group of patients that they called ultra low. So these are patients that basically have the faintest staining on their HER2 tests. So you barely susceptible levels of HER2. That's why they call it ultra low. And we don't have the slide here, but when they looked at those patients as well, they also showed a tumor progression. And that's kind of one of the big advances here, is that essentially any level of HER2 expression, even the faintest level of HER2 expression in HER2 has a benefit. And that was, something that was lauded and talked about as practice changing during the meeting.
Great. Thank you, Adam. Next slide, please. This is the CAR T data, also more data from AstraZeneca. Next slide, please. Angus, will you tell us about what we're seeing here?
Yeah, so I think this is certainly one of the larger positive phase 1 studies we've seen on CAR T in solid tumors. This particular product, what they're calling CCAR31, is a CAR T therapy for liver cancer, advanced liver cancer. And what's impressive here is this high confirmed PR rate, this high confirmed partial response rate. I think in the past, we've seen a lot of CAR Ts attempted against solid tumors. You see basically no efficacy at all. And there was also always this worry that when you use a CAR T in solid tumors, your targets are often going to be targets that might be expressed in other healthy tissues. AstraZeneca here, along with their partners at AstraZeneca and Zhejiang University School of Medicine, they managed to get this good response rate and also what appears to be a manageable toxicity profile. Now, no neurotoxicity in the highest dose level here. You see only 1 grade 3 CRS. So I think also considering that this is a pretty difficult setting, the 91.3% disease control rate is also impressive. We'll need more data to confirm just what is working here about this next generation CAR T and if it'll continue working. So that'll be like what we've seen in the phase 2, phase 3 trials. But one of the key innovations, I think, is the use of a strategy called armoring. This isn't new. Other people have used armoring in the past. And it's basically genetic modifications to the cell that make it more durable in the hostile, highly suppressive tumor microenvironment around solid tumors. I think the hope is that this is going to be a sign that these kinds of next-gen CAR Ts, are finally hitting solid tumors with some success. And I'm definitely looking forward to seeing if that holds up in the phase 2, which is currently ongoing.
Thank you, Angus. Let's see, we have another slide. We can move to the next one. This is the study in small cell lung cancer. Next slide, please.
So really simple here. This combination extended life makes people live longer. This is small cell lung cancer. This is not an indication where a lot of companies have been developing drugs. One of the really clever things AstraZeneca did is that they took their immunotherapy into a bunch of cancers where other people weren't doing studies. And so it's given them those markets. There's a lot we could talk about how clinical trials work and what you do when only one drug has been tested in an indication, but the reality is this helps them commercially and also proves that this PDL1 can benefit patients. So it's just, and yes, the CAR T was also AstraZeneca. There was a lot of AZ at this meeting.
Next slide, please. Angus, we have a lot of questions, so I might ask us to speed through these next couple, but can you tell us on the next slide about the AI patient navigator?
Yeah, so the next 2 studies that we're going to be talking about, or that I'll be talking about, are really about sort of patient care and sort of improving sort of patient experience. This one was about an AI navigator that helped patients reschedule missed colonoscopy appointments or missed colorectal screening colonoscopy appointments. Patients miss these for various reasons. Could be barriers like cost or transportation. And typically, a center might use human patient navigators to help them overcome these barriers and reschedule that appointment so that they get their cancer screenings. That's really hard to do. Thousands of patients will miss their appointments a year. And the idea was maybe an AI could sort of lessen the load on these human navigators. The idea for this at Montefiore Einstein Comprehensive Cancer Center was use this conversational AI to just call the patients and move them over to a human who can reschedule that appointment and maybe solve any barriers that they might have, like cost or transportation. And it worked. A lot of the patients engaged, over half engaged with the AI, and of those, over half of those transferred over to a human navigator, which made it much more likely that they were going to actually finish their colonoscopy. And among sort of the no-show population, which is a particularly hard one to reach, that completion rate nearly doubled. So I think this is just an innovative use of technology to try to get at sort of a difficult care setting, particularly when you think about the population that Montefiore Einstein serves, which is a population with a significant number of vulnerable demographics. 40% African American, 25% primarily Spanish speakers, significant unemployed population, and the AI was still able to engage with a lot of these communities.
Great. Next
We can go to
slide.
the next. Mhm.
This one on palliative care. Next slide, please. Angus, can you tell us about this one?
Yeah, absolutely. So the last abstract is just the plenary on palliative care. I thought this was really interesting because you don't typically see this topic sort of on the main stage. I feel like, you might expect it to be more sidelines, but I think it kind of represented at this ASCO this emphasis on the art of cancer care or the art of sort of really improving patient experience. And this showed that if you deliver palliative care virtually, it's just as good as doing it in person. And that's really important when you think about the advantages of telehealth. You can be at home. A lot of clinicians said that makes the delivery of care more efficient. So you might actually be able to see more patients and maybe make it so you have less caregiver or sort of provider burnout, which is a big problem, of course. And it's easier for the patient. They can have anyone in the world zoom in to that appointment to act as their support. They can get seen when they feel especially sick, which you can imagine is really important for palliative care. And I think this is a sign to a lot of participants that, we can do better in understanding how the experience of serious illnesses like cancer can be improved for patients, and encourage sort of the use of these types of programs without the fear of missing out on any efficacy that you might gain from, you sort of perceive to have gained from in person.
Definitely, and it's good to see this kind of data, patient care data at the plenary session as well. Let's see, we have quite a few audience questions, especially on Enhertu. Adam, we have one here that says, would the ultra-low patients traditionally have been classified as HER2 and received CDK? How will this impact practice?
Yeah, so most of these patients, if not all the patients, did receive a CDK4/6 inhibitor prior to study entry. So that doesn't change. It's really, the decision here is whether whether when these patients then, when their tumors do progress again, whether to go to chemo or Enhertu. So seeing some polls and talking to some docs, most of the physicians that I spoke to said that they would now move towards using Enhertu over chemo, but with some caveats. And I think one of the things that Ashwin mentioned earlier was this idea is as you move these drugs earlier lines, you have to deal with the side effects. And so probably the biggest side effect with Enhertu is interstitial lung disease, which is basically a severe form of lung inflammation. And 3 patients from the study actually did die from ILD in the study. And so what we heard at the podium on Sunday is, from the discussant in the study, is that if a patient has maybe more faster growing disease or more symptomatic disease, then those patients would go to Enhertu. If a patient has it was asymptomatic or has maybe less disease burden, then maybe you would still use chemotherapy before Enhertu. And those are the kind of I think, decisions that are being made. And those are things that will, in practice, when this gets out there, those are the kind of discussions that I think physicians will have with patients.
So this was a really big meeting for AstraZeneca. Matt, can you tell us a little bit about their dream for meeting 80 billion in annual sales and how this data might impact that?
Well, these data actually definitely impact that, because when you look at what their estimates are, a lot of it's coming from Tagrisso, a lot of it's coming from Enhertu, and a lot of it is coming from Lynparza, which is a cancer drug they make that generates a lot of revenue, but that we didn't hear about ASCO this year. So in addition to the more researchy stuff, which could deliver in that time frame, like the CAR T, they have a very interesting strategy there where these were Chinese data that we saw from a Chinese partner of AstraZeneca, but what they actually did is they licensed them the construct, and they are starting their own study of a similar but somewhat different CAR T that uses the construct, but not some of the other cellular mechanics in the US and internationally. So a lot of very interesting strategies there. So yeah, I think we should probably answer some more of these Enhertu questions we're getting.
Oh, Matt, you just want me to do all the questions.
Yeah, I want to make you do all the work. But I actually have thoughts on some of them. But I think on triple negative, we're mostly looking at the TROP2 antibody, aren't we, Adam?
Yeah, we are. I mean, but I think what's interesting about in the question, so this is not approved in triple negative, but I think that question. So why don't you read the question, Catherine, and we can kind of discuss it.
Yes, it says these data are in HER2 positive MBC patients. What are the implications for T-diagnosed use in what we've historically called triple negative breast cancer?
Right, TXD is that's in HER2. That's the
trastuzumab deruxtecan.
Right.
I see.
We don't want to say that.
Thank
so
you.
I think what's interesting, because the question did come up, I asked it, lots of people are asking it, is if you're getting to a point where you can treat ultra low, like why do we even need to test for HER2 at all? Like if there is no HER2 expression, will Enhertu work? And it's a very intriguing question. At this point, everyone that I spoke to said that you will still test for HER2. Like that is not going away. What's interesting about it is that the tests right now that are used are not very precise. They're a little bit blunt when it comes to determining whether somebody has has just ultra low or no HER2 expression. So the discussant during the presentation, Ian Kropp, he's from Yale, he had a great analogy. He said it's like weighing a mouse on a scale used to weigh elephants. That's kind of where we are now with some of these assays. So what this will probably spur is maybe even more sensitive HER2 assays to sort of get to see, like there will be some sort of lower limit of HER2 expression under which maybe Enhertu doesn't work, but we don't know where that is yet. He also talked a little bit about there has been some preclinical experiments in cell lines where if you knock out HER2, does Enhertu work? And what he said was that, it doesn't seem like there's activity in like the true HER2 null, but it's worth running the experiment. And when I talked to Daiichi about this, they said that they are going to test this. They want to see whether if you take a sort of true HER2 null tumor, a patient that has a tumor that does not express HER2 at all, what impact this will have. So, I mean, I think that's one of those, it's kind of, oftentimes ASCO, there's a lot of questions that come out of ASCO. You see data, and then there's like, what's next? I think that's sort of a what's next question.
Let's see. Are there any other Enhertu questions here we feel like we haven't addressed? Taking a look.
Maybe we
the question on, I mean, someone's asking the question on like why does Enhertu work better than other HER2 directed ADCs? And it's a difficult question to answer.
Since he put his name in, right?
I'm sorry.
Leon Tang. This is the one.
Yes. Right. He's asking. And yeah, I mean, there is this idea that the chemotherapy circulates longer, that's attached to it, circulates longer and able to kill more tumor cells. There's this idea that the linker, the linker, the molecule that links the antibody to the chemotherapy, they just hit the right mix of sort of stability and sort of looseness that allows this to happen. There's deeper penetration into the cells. There's all kinds of theories. I mean, when you ask AZ or you ask Daiichi this question, they have these sort of theories and they don't truly know why. I don't think there's a definitive answer for why, but it just seems to have this better effect, more potency than other HER2 directed ADCs.
The other thing I wanted to ask you all about was the CAR T study, and sort of how you see this playing out within the trajectory of CAR T and what this means for the future. Let's see, I'll go to Angus first.
Sure. So I think that reception of this study, when I spoke with experts about it, was a little bit muted. That's mainly because we need to see more data. This is just a phase 1, so you don't really want to read into it in terms of efficacy too much. But AstraZeneca, they are recruiting for their phase 2. They're very excited about it. I think that if they are successful here, will be a sign that there is a chance for CAR T in solid tumors. And maybe some of these next generation kind of strategies like armoring or even other things that people are working on, sort of more in its kind of creative ways to modify the CAR T cell, is going to be a really big part of that. I think, if we can back up and look at the field of cell therapy overall, I think there's been a lot of progress being made in solid tumors. You have the recent approval of lifileucel from Iovance. I think the brand name is Amtagvi. And then there's also a TCR-T coming from Adaptimmune that has a PDUFA date later this summer that will be really interesting to watch as well. So I think, just looking at cell therapy overall, I'm really excited about the future for solid tumors. And this particular study, I think, is going to be, if it bears out in later phase trials, is going to be, seen as an important step forward for CAR T in this setting.
We have been burned before. There were some really cool data on HPV directed CAR Ts that I don't think ever really went anywhere. So, and I think people want to see data from outside China.
That was my next question was, the study was conducted entirely in China, as was another one. And I know that Rick Pazder made comments on that, Adam, in your interview with him on Friday. Can you tell us a little bit about what he said?
Yeah, I mean, this is maybe, Matt, where we can kind of talk a little bit about the summit thing too, if we want.
Yeah, someone's asked about it too, so we can.
So, before the meeting, so maybe we'll set the stage there, Matt. Why don't you tell people what happened on Thursday with Summit, and then we can kind of go into the China thing.
Summit, which is a company backed by Bob Duggan, who is known as the billionaire who sold pharmacyclics to AbbVie, so has had some success in the past and has also had some not successes since. But they put out a press release saying that that in I think it was head and neck, right? They did.
Oh, was it lung? It was lung. It was lung.
It was lung. In lung, they had beaten Keytruda. Which would obviously potentially be a big deal. It's a bispecific of VEGF and PD-1. And the question is that working better? And there's not much data, but that study is conducted entirely in China.
Right. So the idea is, now Summit is running their own studies of this compound, this bispecific. They're running studies in the U.S. So it's almost a little bit of a moot point because they're not, and they've said they aren't trying to get approved here based on the China data. They're running studies here, and I think off the top of my head I don't remember when those studies read out. If anybody in the Q&A can remind me when that study is supposed to read out, let us know and we'll mention it. But the issue is, the issue there is just the reproducibility, right? Will these results that we've seen that they have reported on in China, and we haven't actually seen any of the data, as Matt mentioned, it just was we beat Keytruda, and that's all they said. Whether those results are reproducible, meaning that if you run a study here in the United States with a broader patient population, a population more relevant to the U.S., will you get the same results? And we don't know, right? I mean, that's just one of the questions that's out there. Now, to be fair, there have been successful drugs that started development in China that are now here. I mean, the one I think of most probably is the CAR T that Legend Biotech developed originally, was licensed to J&J, Carvykti, right? If I'm getting the name right. Started in China. I remember a few years ago being at a meeting of an ASH meeting where early data from that drug, that CAR T, were presented, and honestly, people didn't believe the data. I mean, I remember there was almost hostility in the crowd as people were questioning whether those data were legitimate or not. And they turned out to be legitimate. That turned about that those data were real, and they were reproduced here in the United States in clinical trials, and the drug is approved now. So I think that's kind of, this idea that no data in China is legitimate or reproducible is just not true. I mean, we have seen that happen. So, we'll just have to see what those studies play out. Where we get back into the pastor thing is, because he has in the past sort of been more receptive to data from China, but now the policy here at the FDA, as reiterated by Rick Pastor at our event on Friday, was that in order that I have the quote, I wrote it down. Hold on a second. The quote is, what he says, he's pro multi-regional trials, right? And that what that means is that you want data from a broad region of the world. That's the kind of most representative data that you can get. We don't want data where you get like 90% of the data from one place and maybe 10% from the other. So that's the data that they want. It's why it's the most representative data. It's probably the strongest data. So that's why this idea is like, you can't go to the FDA with just data from a China study and expect to get approved. You need to have this multi-regional trial.
One thing about Summit that I think we all want to explore as a further question is what data exist on using separate VEGF and PD-1 drugs, and has that been tried before? I did speak with Jed Wolchuk, who's one of kind of the gurus of immunotherapy, one of the key figures in developing the PD-1s, and Yervoy, and he said he didn't think that idea had been fully explored. So maybe this isn't as out of left field as it might seem. But I want to test that idea a lot more than I have.
We have another CAR T question. Angus, you mentioned that some of the reception was muted. Leon Tang asks, at ASCO, the overall interest in CAR T TIL for solid tumors seem lukewarm, but there are strong interests in T-cell engager for solid tumors, probably due to the approval of Amgen's tarlatamab and TCE from Janux. What sentiment have you gotten from this CAR T versus TCE rivalry? Does anyone have any thoughts on that?
The sense that I get from a lot of investigators is that these therapies have pros and cons. And I think that, it's not really one or the other. It's going to be both. We're going to need both of them for different reasons and in different settings. I think, CAR T has the benefit of, being this living product. I don't know that we fully understand everything that it does once it's in the human body, compared to like T-cell engagers, for example. But I think bispecifics, specific T-cell engagers, have enormous logistical benefits over CAR T. CAR T is hard to manufacture. There's very limited production for it. And bispecifics can be made much more easily. They can go to much more places. You can administer them in more rural settings, for example. And they're in general tend to be, I think, a little bit safer than CAR T. Although I think now the CAR T toxicity that we see is very manageable. Expert clinicians have really figured out how to, treat things like CRS, high-grade CRS with antibodies like tocilizumab. So I guess that's what I would say. I don't know if Matt and Adam, you have other thoughts.
Yes, no.
I think people would rather not use a cell therapy if they don't have to. So I think that.
Yeah, I think that's why there is sort of this kind of buzz or excitement about TCEs, right? Is that you avoid having to go the cell therapy route. And so yeah, that's kind of where that's where I didn't see any TC data. I didn't get a chance to if there was any at ASCO. I didn't get a chance to see any.
I didn't see too much either, yeah.
A really helpful comment from the Q&A that someone made. In fact, this person should get credit, and it's Bertrand Delsuc. Is that Destiny Breast 15 is checking that question we were just asking about super low. So that's the trial for you all to look up if you want to watch one.
Bertrand is on top of everything.
Yeah,
Follow him on Twitter.
it's truth. Adam, do you have any questions for us?
Do I have any questions for you? Well, I don't know. Maybe Matt, you and I we should talk a little bit about the IRA and the impact, because that's come up a couple times. You and I went to this pharma being the industry lobbying group. We went to this press conference that they had, and Gilead CEO was there, Merck CEO was there, Lilly CEO. What was your takeaways from that with respect to kind of cancer drug development and the IRA? What did you hear from the CEOs up on stage?
Well, I mean, my favorite thing was just that we got the fact of how much Merck has spent developing Keytruda, which is in our new. I think it was like 46 billion. That was really cool. I've been meaning to say a little bit more about this though, which is that look, there are a lot of good points being made about the IRA, and the fundamental problem with the IRA, and I think with a lot of our attempts to control drug prices in the US, is they create these market distortions, right? Like the big one right now that the drug companies all mention is that small molecules get a shorter exclusivity life under the IRA before the IRA kicks in on them than biologics. And this is obviously dumb from a big picture perspective, because small molecules go generic and get really, really cheap, and biologics go generic and get 30% cheaper. So for cost reasons alone, you'd want to push drug companies towards small molecules, not away from them. And there are other distortions that are built into the law, but they also say, like, I asked David Ricks about this, because the problem is, like, yeah, there are distortions, but there are distortions partly because there's never been a real consensus on how to fix the US market so that you do get better control of drug prices. And there's a sense that cancer drugs have gotten too expensive, that Keytruda is way more expensive than Taxol was, and is that justified? And I think that's the problem they always have making this argument is that, I mean, what Rick said is that, well, drugs aren't the main contributor to healthcare pricing, which is true. Hospitals and lots of other things contribute, but they always seem like it's like, well, yeah, we could give up a percent of growth or something, but we can't. So I think it is kind of hard, even though there are these problems with the law, that it doesn't seem realistic that the government's going to accept not taking the price cuts, which the government.
Matt, what did you think about the idea? Because they talked about this idea that it disincentivizes them from testing drugs in the earlier stages of disease.
I think that's absolutely true.
What do you think about that?
I honestly think that it probably does, that it used to be, because now what happens is if you bring out a drug in a rare indication, and you were thinking you'd have a bigger one. Tagristo's actually like the best example. Tagristo's first approved in like advanced lung cancer, and now we're seeing these trials in adjuvant, right? Where the market got much, much bigger as they went up those trials. And what they're actually threatening to do is not do the studies or not get the drugs approved anywhere else. They're saying, well, we just won't file for approval in the US until we've got a big enough market. I could imagine them doing that, but you do have the push and pull of their patent is burning that whole time, right? So like,
we asked Pastor about this on Friday night, and again, he's a FDA official. He's sort of supposed to be apolitical, so he was kind of. I think he didn't really want to answer the question, but he did say that that he would prefer companies develop drugs earlier than later. And he
Right.
said, there's no reason to develop them later before you develop them earlier. And that has been the model, right? I mean, traditionally, we see drugs get approved, developed and approved in later lines because those studies are shorter, they're easier to do. It's where you can get accelerated approvals. So that's what companies have done and then moved to earlier lines of treatment. And what Pazder was saying was, hey, I would actually prefer you do studies in earlier lines first, or do them simultaneously or something. So I thought that was kind of an interesting comment from him.
So I brought that to Susan Galbraith from AstraZeneca, who runs their R&D, and what she said is, well, I basically did that with Tagrisso, that she was told they did phase 1 studies in earlier line cancer at the same time as they were doing later line. But we still are looking at, like, if you look at this drug, like this is a drug that was approved in 2015. We saw the Laura study this year, right? It's been 9 years. Like it just takes longer to do those studies is the problem. And if you think your big market's going to be adjuvant, and there are a lot of forces in the drug industry, this has always bothered me. Like there's always the question in high cholesterol, right? Like if you could develop a drug for familial hypercholesterolemia, which is rare genetic high cholesterol, or like regular high cholesterol, there are instances where you're just better off doing the rare disease and charging a ton of money and calling it a day and not helping everybody. So there are structural problems that have kind of been built into the US system that encouraged high price, smaller market drugs over somewhat lower priced large market drugs. But when companies can get the big market, look at the GLP-1s, look at Tagrisso, they most certainly do want it. So.
Well, Matt, you had at our event on Friday night, we had Pazder on stage, and that was fun. But you also had a great panel, R&D panel. You mentioned Susan Galbraith was on the panel, Ken Keller from Daiichi was on there, Levi Galloway. So tell us a little bit, like what do you think the highlight was? What was the headline out of your R&D panel on Friday night?
God, it's so hard to remember.
I know, it feels like it was weeks
ago. But I thought like a big highlight was talking about there was this really interesting question that Levi Garraway kind of asked me on the prep call, Levi Garraway from Roche, which was, do we think that are we in an era where for a while it was all about tyrosine kinase inhibitors, and then it was all about immunotherapies, and then it was all about antibody drug conjugates, but now there is a little bit more of a sense of all these modalities are working and they're all being used. I hate the word modalities, but all these different ways of making drugs, and we're seeing different and we're seeing kind of different thoughtful approaches to each. I mean, if I had to pick one that I actually think the TKIs they just keep working. And then we saw the Crown data on Lorbrena from Pfizer and the Tagrisso data, like just when you have a good target with a TKI, it almost always works. In immunotherapy, we really haven't gotten that much in terms of new mechanisms. And my question for him, which I don't feel like I got a great answer from, but there's a lot of people who seem to think that we've kind of cracked the antibody drug conjugate thing with Enhertu, but antibody drug conjugates have been really finicky. So I wonder if we're going to see as much success as people are expecting, or if it's going to kind of be between TKIs, where it seems like every other one works, and immunotherapy, where it seems like after PD-1, I mean, we did get LAG-3, but it really seemed like a lot of the immunotherapy targets haven't worked. So
I mean, there's a ton of ADCs. Like you can't keep track of all the ADCs out there. There's like, there's so many, with different antibodies, and different antibody targets, different chemotherapies, different linkers.
Yeah.
I saw a little company, Corbis. I saw their data. I wrote about them because it's the second best performing stock this year. They have a Nectin-4 targeted ADC for head and neck cancer. I'm sorry, not for head and neck cancer. What is it? I'm sorry. No, bladder cancer. You know, again, and it came from China. They licensed it from China. We're talking about China, and the data that they presented at the meeting was from a study conducted in China, and they're doing their own U.S. study. I mean, but that's just one example, right? There are just so many. There's just dozens and dozens of these ADCs out there right now. It's so hot right now.
We do have an ADC question, which is ADCs are all the rage and they're super expensive. Would there be biosimilars of ADCs in the future? Would it be the IRA or a biosimilar that could drive any price reduction in the future? I don't know if anyone has thoughts on that.
I mean, theoretically, yes. I mean, the answer is yes. I mean, we do have, we have biosimilars to antibodies right now. As Matt mentioned, they are not priced at the same discounts as a small molecule. And they may be, and the complexities of ADCs manufacturing may be such that making what would be sort of a biosimilar ADC might be really difficult. I don't know enough about it to answer that question, really accurately or not, but you could see where that would be that might be a difficult thing to do.
And Leon Tang also wrote, some people argue that the next big theme is to upgrade current PD-1 chemo and many indications with PD-1/ADC for fancy chemo. What is your view on this thought?
I would do it if
I mean,
were a company.
Yeah, I mean, I think the idea there is that, the potential to get better efficacy and maybe even better tolerability and safety by using a checkpoint with an ADC versus a checkpoint with chemo, right? And those studies are not, I mean, those studies have been done, and there's studies that are done. I'm thinking like the one that's on the top of my head, just because I was reading a note about it, was, Gilead is testing Trodelvy, which is their Trop-1 targeted ADC. They're testing it in combination with Keytruda versus I think it's worth Keytruda against Keytruda alone in lung cancer, right? So the idea is, right now Keytruda and chemotherapy is a standard of care in lung cancer, and what if you used an ADC and Keytruda together? Would that provide better PFS benefit, better OS benefit, and we'll know at some point.
I just answered a question in the Q&A, but then they said it was actually meant for Angus. So I want to ask Angus, do you think that first generation TIL therapy will be a modality that will be adapted widely in the solid tumor space, given the burden required of the burden of the required protocol to deliver this treatment, lymphodepletion, the AE profile?
I'm going to take the slightly cop-out answer here. I don't know, Matt, if you had a better one, but it is sort of like time will only tell. I wish I could predict the future, but I can't. One thing I would say, though, is you're right. Lymphodepletion is hard on patients, and you have to be healthy enough to go through it, right? That's always tough. And adverse events, always a concern when it comes to those kinds of things. I think TIL therapy, look, the actual TILs themselves don't look super toxic, at least when I was looking at the data. And the same is true for all the heme cancers that have CAR T indications, right? Like the people died from those CAR Ts, but they are in high demand. I think that for TIL, the first generation of TIL, the question will be, what can you get out of it? What is the benefit really going to be that we're going to see in the real world? If it's going to offer people like lasting remissions, total remissions, then for some patients who fit the profile that they can take the prep for it, sort of the lymphodepletion that you talked about, then yeah, maybe I would expect that clinicians might be enthusiastic about it. But it is a very new approval, so I haven't really followed
I think,
it too closely.
I think the only thing that brings up too, Angus, is it's so hard, these meetings. There's so much going on. Like, Iovance did have some data right at the meeting. I didn't get a chance to look at it. I just ran out of time. And that's what happens. You go to these meetings, and it's so difficult to kind of keep track of everything when you're there. You're running around. There's multiple things going on. And so I didn't get the chance to see the eye events data. I mean, another one that we haven't even talked about, which I didn't go see the data either, was the Merck Moderna, their mRNA vaccine for melanoma, which they had durability data, like I think it was 3-year durability data presented. I guess they're not calling it a vaccine, they're calling it an individual neoantigen therapy. I think that's the buzzword that they've used now for that, which is a fascinating therapy. And Merck and Moderna are really excited about that in melanoma, and then they're looking at it in other cancers too. But I didn't get a chance to see those data at the meeting.
The data are good. I mean, there seems to be a little bit of a debate about whether that could have potential for accelerated approval or
Yeah,
whether wait for full data, and I think that's
right.
probably depends. But both those data and also the BioNTech cancer vaccine data have so far been promising. So like that whole mRNA for cancer is a
Yeah, I mean, it really does seem like coming out of, and I saw some comments, more comments from Moderna than Merck, and you can understand why. I think Moderna is probably, for a lot of reasons, wants to file for accelerated approval with that INT, just because they need something new other than their COVID and RSV and other vaccines. But it does sound like they're really going. I mean, it sounds like they're going to file. That it's really more a matter of time. One of the things they have to wait. I think they're going to wait for full. They're enrolling a phase 3 study now. I think they'll need to wait for the enrollment of that study to be completed. Again, that's another FDA thing. FDA really wants these phase 3 studies, the confirmatory studies, to be well on their way before you file for accelerated approval. So I think they need to do that. And I know that they have some getting the manufacturing facility for that product, get that completed and inspected. I think that's another kind of gating factor. But it really did sound like they're going to file for accelerated approval for that INT in melanoma.
there's no downside, obviously, at that
Yeah.
point.
Yeah, and that phase 3 study they're running, that's a long study. I mean, you're talking about like you're going out like 2029, 2030 to finish that study. So it's a long time before you'll get data from that study. So again, that incentivizes them to file for accelerated approval.
We only have a few minutes left, so is there anything we haven't gotten to yet that you all took away
Any
from
other
the meeting?
questions in there that we should really answer, Catherine? Anything that's not on these topics these drug trial topics that we've hit, maybe?
Let's see.
There was one on AI modalities in oncology practice. I don't have a great answer for it, but maybe we should ask that one.
Oh, then you take it, Matt.
Okay, I'll read it. Can you comment upon the fact that the main section of AI modalities in oncology practice field showcasing the advances in prostate cancer for artificial vision and the recommendations around predicting response to systemic treatment, which took place in the main hall, was very poorly attended? This session presented disruptive technologies of multimodal AI and a framework that will most likely become tangible very soon, but maybe the clinicians are still not ready to adapt this technology, in parentheses, in denial. Angus, what do you think? Why didn't people go to that session? Are this because physicians are in denial?
I thought you were answering the question, Matt. You just read the question.
I think physicians are not in denial. I think companies that think physicians are going to be fast I think that the AI that physicians will be fast to adopt is the AI that makes physicians' lives easier. Like we had a really good demonstration of note-taking software at our Stat Summit in San Francisco. I think some of those other advances are going to be hard and are probably going to be driven as much by payers as physicians. So yes, the physicians are in denial, but nobody's more in denial than people working at a startup until suddenly the world changes and they're right.
Angus, do you have a different answer?
I don't. I actually I missed the question a little bit because I was reading some of the other questions on the Q&A.
It was why didn't people go? There was a big AI and prostate cancer session apparently in Hall B1. It was very poorly attended, and so are clinicians in denial was the question.
Oh, I see. I mean, I think I agree with your answer there, Matt. I mean, I think there's a lot of uses for AI. There might be, I actually kind of, part of me kind of wonders if there's a little bit of AI fatigue that like you see it all the time now. It is it's still obviously a very exciting thing, but it there's a lot of it.
Also a lot of
There's a
AI,
few,
but not a lot.
there's a few questions on radio pharmaceuticals. I didn't see any radio pharmaceutical data at this meeting. I didn't get a chance to. I don't know if anybody else saw any. Sorry, I guess that's not a great way to answer the question, but I didn't see any. Matt, did you see any radio pharma data?
There wasn't, I may have, but there's not anything that that leapt out at me from.
I'll tell you what though, Novartis.
I remember talking about alpha particles, and I'm trying to remember which.
Pluvicto, which is Novartis. That's Novartis's, right? That's their radio. There was a lot of advertising around Chicago.
We should do a little bit of just color here. The scariest ad I saw the whole time is Novartis had an ad for Semblix.
Yeah, the bull.
In the riots.
Yeah.
There's this giant bull running at you, which after a couple drinks, that bull looked pretty imposing.
Yeah, Novartis spent a lot of money on marketing at ASCO this year. They were all over, a lot of ads.
I saw a Sanofi ad that referenced Clash of the Titans that I really liked, and I tried to take a picture of it, but it was on a video display, so I couldn't. Just came off as a bright light on my phone.
Yeah, but anyway, yeah, sorry, I did not see any radio farm or radioligand data, so stay tuned. Well, I mean, we do write about it, so we'll have more, but yeah, I didn't see anything.
Adam, did you want to talk about the Novartis drug that you had some suspicions about?
I mean, people probably read it. I don't have anything more to say. And it wasn't at the meeting, but I mean, it was, but read my story about why that was a very bad deal for Novartis to do.
Do you think there's anything Novartis can do at this point, though, Adam? I mean, they've bought this thing, right? So don't they just have to write it out, and it's a $3 billion deal?
They have to figure out what kind of data they need that will allow them to file. We're talking about pelabresib in the morphosis drug in myelofibrosis. Yeah, I think what I hear is that they're consulting with experts and they're trying to figure out what to do. Yeah, and what data they need to collect, where that data can come from, and whether they have to do more studies, and it's stuff that they should have done before they decided to spend $3 billion on it, but they didn't. So there you go.
I think the other thing to note, which we were talking about in the call here, someone asked about marketing. I don't know about a good marketing gig, but I was impressed by the diversity of food offerings at the booths on the exhibit floor this year. Lily had Italian beef, and Isai had guacamole, and I think somebody had juice shots. I mean, it wasn't just Dippin' Dots and
but this is just a way to buy our influence, isn't it, Matt?
Yeah, but it's nice that people are doing it in different ways, and that it's not just
food options. The food options are bad. Like the regular food options are bad. So that's why it's another this insidious influence of big pharma. When we I think that was that's my
Well, yeah, no, the food options in convention centers are bad. That is true.
Yeah, it used to be. Remember the old days when you used to get golf clubs? There was there was nothing. There was no limit on the number of gifts that you would get at a big pharma convention booth.
And then for a while they'd have signs that physicians from certain states were not supposed to get the ice cream.
Right?
They still have those signs.
Yeah.
They were on every sort of hospitality station. They're if you're from Minnesota.
Food must be exempt because there's a lot of food now down there.
This is someone asked about the recording for this, which I think we're drawing to a close here. But
yes, we are.
The recording. If you signed up to attend, I think that you will get a link to that. I don't think it's limited to stats. Oh, there's an easy answer to this. I tweeted out the link to watch this live on our site. That link, I believe, is not paywalled. And we'll have a recording of this immediately afterward. So just go to Twitter.com/Matthew Herper, and you can relive this entire experience.
Yeah, the fact that we're talking about food is probably a good sign that we should just.
Yeah. Well, okay. My closing question for each of you, very briefly, is what do you want to see next year, either science or food? I'll start with Matt.
I really do go to ASCO for science, not food. I did feel I don't have a specific drug that I'm like waiting to see at ASCO next year, but it would be, and like the mRNAs were a good example of something new. I was really excited to see the Lorbrena data. I was really excited to see the Tagrisso data. I thought the Enhertu data were really exciting, and ADCs also represent something new. But it does strike me that I've been covering PD-1s for 10 years. I've been covering TKIs for 20 years. I'd like to be blown away by something I haven't even thought about writing before.
Angus, what do you want to see?
That was a good answer from Matt. I think that I'm excited to see more cell therapy data. I think cell therapy is really interesting, even though it is hard to give. I'm also really excited to see more data on patient care, really. I think health services, I've said this before, but I think health services is a really important part of sort of the experience of cancer patients in the healthcare system. And I want to see more data on how that can be improved and how the health system can be made more efficient. So it's cool to see that this year, and hopefully it'll continue.
Great. And Adam, do you have dreams for ASCO 2025?
Yeah, just a bigger venue for our event next year, because we're going crazy with Rick. Maybe we'll have Rick Pazder on again, because he's never going to retire, right? The guy he's riding off. He's going to die in that office. So hopefully he doesn't before next year, right? Of course not.
Hopefully.
It's a picture of Irving Health. No, of course not.
The events team is sitting here DMing me like,
yeah, I know. We love Rick. We want more Rick at our event next year. That's what I want. I want more Rick Pazder at our event next year.
Great. All right. And
awesome. We love him.
And with that, thank you everyone for joining. Thank you to Angus, Adam, and Matt for answering these questions. Thanks to our event sponsor, Precision AQ, and to all of you out there for taking the time to join us. Our next upcoming annual flagship Stat Summit is taking place October 16 and 17 in Boston, both in person and virtually. We will gather top industry leaders. The theme will be on how to change medicine, even though for the better, even though it's hard. And more information about that can be found on our website. So thank you all again, and we hope to see you back next year.
Hey guys, get some sleep. You can tell I need it. I'm a little punchy.
Yeah.